So we've had some great talks, and thanks to the previous speakers for those talks about the different NAMs, different tools and approaches that might be important in NGRA. So now we're going to turn our attention to how we might integrate the information that we've heard about so that we can make safety decisions for cosmetic ingredients. Now, of course, animal-free approaches have been an important feature of cosmetic ingredient safety assessments for a long time, mostly for assessing exposure or local effects. And at the moment, we can make animal-free safety decisions for systemic effects depending on the level of exposure and the similarity to existing molecules which have systemic toxicity data by using approaches like exposure-based waving or read-across, those sorts of approaches which are at the low tiers of the SURAT1 workflow. But the major gap that really exists is a way to confidently assess the safety of a completely novel substance where it's not possible to use these lower tier existing non-animal approaches. And I'll be talking about one particular way of integrating data today, but an important thing to say at the outset of this talk is that there are many different ways to approach safety assessment. There's no one-size-fits-all NGRA, and a lot of the tools and concepts that we've been talking about and that I'll be covering in my talk really do need scrutiny and need evaluation so that we can understand their strengths and limitations and so that we can understand how far they take us in decision making. And actually that's why the ICCR principles that Kate shared right at the start of this afternoon really is so important, because these provide us with the anchor that remind us that what we need to achieve as we start to build a bespoke NGRA using different kinds of data is going to be flexible and fit for purpose. Hopefully these different principles help to keep us honest in terms of the strengths and limitations of the different approaches that we're going to be using and applying in day-to-day decision making. So the shape of the NGRA that I'll be talking about, which I think is achievable in the short term, is one that's been set and championed by the US EPA in the context of smarter prioritization of substances. Now, this chart is taken from a milestone paper from the EPA, which looks at a comparison between predicted human in vivo exposures and a whole range of in vitro points of departure values, which have been converted to an equivalent human in vivo daily dose. So you can see the exposure, predicted exposures here with the green boxes, and the range of in vitro AC50 values which have been converted to human in vivo daily doses up here. I don't want us to focus too much on the substances here, but this type of plot nicely illustrates what a relatively simple comparison of in vitro biological activity and human exposure might look like. In this instance, the points of departure, well, these all come from ToxCast assays, and the exposures are estimated using high throughput toxicokinetics. So this really is a high throughput prioritization approach where you have a large margin of exposure, so a large difference between the oral equivalent point of departure and the expected human exposure. That would indicate a low priority for follow-up, but where you have a smaller margin, maybe like this one here, or where that's overlapping here, that would indicate a high priority for follow-up testing. And the important thing about this approach is it doesn't attempt to predict what adverse outcomes or pathologies might be associated with exposure to these substances. It's simply based on the premise that if the in vitro assays provide enough biological coverage across the different modes of action that could lead to adverse effects, that large margin of exposure would be protective of human health. So in other words, what we're talking about with this type of approach is protection, not prediction. Now, the assumption that the points of departure from these in vitro tests are at least as protective as the points of departure from in vivo tests needs to be challenged and tested before we can be confident to use that in safety decision making. And several regulatory authorities are doing just that. So on this chart here, we've got a comparison of in vitro and in vivo points of departure. And what this comparison, if you haven't read this paper from last October's issue of Toxicological Sciences, I'd strongly recommend that you do it, because what this shows is that for 92% of the chemicals that were evaluated in this exercise, the in vitro point of departure was more conservative than was the in vivo point of departure. And it's analyses like this, and hopefully more analyses that we'll see coming out of this APCRAS initiative and other initiatives as well, that are going to really pave the way for the reduction in animal tests and hopefully completely a fully replacement, as Chris was reminding us earlier. Now, as I mentioned earlier, the context of this work is currently on using NAMs for prioritizing substances for testing. But what about using these same approaches in a different context, in exposure-led, risk-based decision-making according to the ICCR principles? What we need to know is if this approach of protection, not prediction, is going to be helpful and where it falls down. So to answer these questions, within Unilever, we've been performing a number of different NGRA case studies on well-known ingredients. And we've been imagining we had no existing toxicology data and no presumption of safety. And this is just one example that's been published recently in Toxicological Sciences. It's a safety assessment for coumarin in body lotions and face creams. And just to make it clear, this work wasn't done to assess real consumer products that contain 0.1% coumarin. These are hypothetical products, hence the less than slick homemade labels on the bottles. So a case study needs a framework, and this is the overall framework for the case study. Now, the path that was followed for this case study is shown on this slide, but as I mentioned earlier, the same approach may not be right for everything. But just like the SURAT1 workflow that Kate mentioned, this framework does start with exposure. It considers all the existing information available on the ingredient, and of course, in this case, carefully removing any existing animal data. And then it moves through to characterizing the in vitro biological activity of the ingredient, and it considers what refinements are going to be needed and then finally arrives at a safety risk assessment decision by the calculation of a margin of safety or margin of exposure. Now, the exposure information that's needed right at the start of this framework includes habits and practices data, as Corey was mentioning earlier, so we can work out how much of the product and therefore how much of the ingredient consumers apply to their skin. It includes in vitro data on how much of the ingredient penetrates the skin over time, and importantly for PBK modelling, how quickly. It includes in vitro clearance data and other parameters needed to build the PBK models that Corey was describing, so that we can understand systemic exposure to consumers following use of the case study products. And that internal estimate is really important because it drives the risk assessment, and along with the measures of in vitro bioactivity, it's what enables that final margin of exposure or margin of safety calculation, so that we can arrive at our overall conclusion. Now, this is just some of the output of the PBK model that was built for this case study, which you can find described in much more detail in this paper by Moxon et al. It's important to note that the PBK model was built in a tiered manner. So everything about this assessment is tiered. Everything about NGRA should be tiered according to the ICCR principles. And development of the PBK model started with in silico predictions. And what you can see on these charts here is that the key parameters like hepatic clearance, where you use a in silico prediction for that, you can see that you get a very conservative estimate of plasma exposure compared with when you use a in vitro derived input. And using this model, we're able to calculate exposure over time and also to determine the key metrics for the safety assessment, which in this case was the maximum concentration in plasma across the population following use of the case study product. So moving on to the in vitro bioactivity data that we used, a key piece of information was from an in vitro safety pharmacology screen. This was conducted by Eurofins on 44 cellular targets, covering a couple of nuclear receptors, G protein coupled receptors, ion channels, enzymes, and transporters. And these targets were originally defined in this publication here by Bowles et al. As key reasons for safety-related drug attrition. So they're a good starting point for a cosmetic ingredient relevant panel, but not necessarily where we would want to end up. As you can see in this chart, all 44 assays for coumarin were negative. Now, I don't need to go into this in too much detail because you've already heard from Paul today that potentially an important mode of action associated with toxicity is the induction of cellular stress. And Paul's already told us about the panel that we've been jointly developing to characterize changes in key cellular stress pathways. So the eagle-eyed among you will have noticed on Paul's charts that coumarin was run through this panel. And one of the neat things about this work, which you can read about in the paper that Paul mentioned, is the Bayesian statistical approach that was used to interpret the data and provide the points of departure. Now, this modeling approach was applied to the whole data set to find points of departure and to explicitly quantify the uncertainty around these points of departure. And although coumarin wasn't particularly active in the cell stress panel compared with some of the other substances like Paul was mentioning, diclofenac, doxorubicin, we were able to derive a point of departure for possible effects on these cell stress pathways, and you can read more about that in the paper. So finally, a really important technique used in this case study was whole genome transcriptomics using the BioSpider TempoSeq platform. And the reason these data are so invaluable is they look at changes in gene expression across the whole genome. So they provide us with broad biological coverage. This case study used 3 different cell types, which can of course be expanded to increase coverage and confidence as needed. Now, transcriptomics data can be used in various different ways in the safety assessment. So for example, you could use it to identify modes of action driven by gene changes, or you could use that information to help to substantiate substance groupings for a read across. But the way that it was used in this case study was to identify a no observed transcriptional effect level, or NOTEL. The assumption here is that many adverse effects would result in a transcriptional change. Therefore, keeping substance exposures below the levels that affect gene expression is going to be protective for human safety. NOTELs can be derived in several different ways, either at the gene level or at the pathway level. And this table gives the NOTELs for some of the different analysis methods used. Because a change in gene expression doesn't necessarily result in an adverse effect, using a NOTEL in a safety assessment alongside all the other functional assays that we've discussed is probably going to be by its nature quite conservative. So in other words, if consumer exposures are greater than these NOTELs, this doesn't necessarily mean this is going to cause an adverse health effect. But if you're in that sort of situation where your exposure is exceeding your NOTEL, you probably need to dig a bit deeper into the mode of action, work out which pathways are being affected, which genes are being affected, and therefore what that biological activity could mean in terms of an adverse effect. And this means that the NGRA would need to flip from sort of lower tier protective, not predictive mode that we've got described in this framework to a much higher tier and much more ambitious predictive mode. So one of the questions for this type of protective, not predictive NGRA is how useful it really is. For intentionally applied products, how often do the in vitro points of departure give you a safety margin that gives you reassurance of safety? And how often do they give you a safety margin that drives you down a hole of digging and digging and generating more data? And that's one of the reasons that case studies are so important, because we can only really answer that question with more examples of end-to-end next generation risk assessments. Now, there's a lot more data that sits behind this case study that I haven't touched on. You can see that on this schematic here. So please do take a look at the paper for those and see how we've handled some of the 3D models and some of the metabolism refinement. Now, of course, a safety assessment requires a margin of safety to be calculated. This is what the final margin of safety or margin of exposure plot looks like for this case study. As you can see, the yellow line here represents the plasma Cmax and its associated distribution for the face cream, and the purple line represents the body lotion. And the points here all reflect the different points of departure and distributions for the different in vitro assays. So that's good news for this is that the dots are up here and not down here. these are the final margins of safety values, and you can see that they're all well over 100. Knowing what we do about the relative conservatism of in vitro versus in vivo points of departure, and also the fact that this is an internal margin of exposure. So in other words, the kinetic differences are already taken into account in this safety assessment. These margin of safety values look quite encouraging, I would say. But an outstanding question for NGRA is, what margin of safety is needed to demonstrate safety? And I think rather than a magic number to shoot for, an appropriate margin of safety is going to be dependent on the assays used and informed by benchmarking to existing substances, as Paul was describing earlier, with a known safety profile. And I think most safety assessors would regard these as fairly large margins of safety and potentially giving you the ability to be able to make a safety decision. So in conclusion, we are able to integrate available tools to make a non-animal safety decision, but we have some way to go to understand how protective the resulting safety assessment is. In other words, we need to work out how confident we can be in this type of assessment. We need to view NGRA as a flexible approach, which is tailored to the decision context, tailored to the decision, the question being asked. And the approach I've described here is protective, not predictive. But depending on the assessment question or the outcome of that initial comparison between exposure and in vitro bioactivity, the assessment may need to flip into more of a predictive mode. We've discussed several new in vitro and modeling techniques, and if we're to confidently use these in safety decision making, we need to make sure they're robust, we need to characterize their associated uncertainty, and we also need to rethink what an appropriate margin of exposure or margin of safety looks like with these new tools. Now, these aren't small tasks, and we know there are many shortcomings. We know there are uncertainties with this approach that need to be ironed out and addressed with more research and more discussion. But with that research, with more creativity, with more examples, I'm sure that we can start applying this to more and more exposures to cosmetic ingredients. So this work has been supported by the great team at Unilever, as well as the partners that I've mentioned, but a special shout out goes to Maria Baltazar, who did a great job of leading on the Coumadin case study and shepherding the team to get to the point that we've arrived at now.